Hey guys, so we're studying asexual reproduction in plants this week, or today, more so. So get out your notes and label these asexual reproduction, and I will highlight what you need to know, okay? So rhizome. So what is a rhizome? It's an underground stem. So the cool thing about rhizomes is this is your plant on top. Okay, so the rhizome is this, okay? So it's an underground stem basically. So how does it work? Bulbs are produced at the nodes to develop branches that stay underground, okay? So if you can look, and I'll show you on the next picture to show you what a rhizome actually looks like. So some examples are grass, cattails, and asparagus. So basically what happens is right here where my highlighter is, these are your nodes. Okay, so it's basically what happens is when you pull it, it's not necessarily all going to pull up, but you will have, wherever you have a node, you're going to have a new plant. That's why grass does so well. So tubers. What is a tuber? It's basically a modified rhizome, okay, because you have your potato plant, it's right here, but your tuber is actually your potato. So how do tubers work? Their specialized stem branches grow down into the ground as well as swell up with starch-containing cells. So that's why potatoes are very starchy. Okay, so some examples are Irish potatoes and sweet potatoes. So here you go. So this is what a potato looks like. If we left it like this and we planted it, it would grow from these spots right here. Okay, so those are going to be your tubers. So runners. So what is a runner? It's a horizontally growing stem that runs or creeps along the ground. So here's your runner, okay? Here's your runner. So this is our first plant, and it ran basically down here and created this plant. Let's see, how do I get rid of that? Okay, it doesn't let me get rid of that. Okay, so how do runners work? New plants will grow at the end of the runner. Okay, examples are wild strawberries and spider plants. And I'll show you on the next slide what the runners look like. Okay, so this is my runner. So this was my first plant. This is the plant that I planted. Okay, so then my runners are these right here. So then a new plant, then it ran a little more, then a new plant. So basically it kind of looks like strings. So cuttings. What is a cutting? It's a vegetative piece of the plant. So maybe a leaf or a piece of the stem or something. Okay, it's going to be vegetative. And see what happens is they put the plant in, or they put a leaf of the plant right here in the soil, and it'll grow. So it's considered a cutting. So how do cuttings work? A cutting from a stem, a leaf, or a root can be planted in the soil or other growing media. Okay. And it'll eventually develop roots. This is not going to happen overnight. It's going to take some time to do. Okay, so examples are coleus, geraniums, and African violets. So you take your cutting, you put it in some rooting hormone, and then it will grow a new plant. A bulb. So what is a bulb? It's a short, swollen stem that is surrounded by fleshy leaves or scales. Okay, so this is what a real bulb looks like. So how does a bulb work? A shoot apex grows from the nutrients stored in the leaves. So basically the leaves already have some of their nutrients. And then some examples are onions, okay, chives, and lilies. That's why lilies come back every year. And this is what a bulb looks like. You just plant it in the ground. So corms is a short, swollen stem without storage leaves. Okay, so think about a bulb has storage leaves. Okay, this one does not. It kind of looks like they're stacked on top of each other. Okay, so how does this work? Each year a new swollen stem will grow on the old swollen stem. So it continuously grows. So it's not a bulb, because if you pulled it up and looked at it, you would be able to see how they stood on each other. So examples are crocus and gladiolus. Okay, so you see a bulb is just going to stay one. Okay, this is stacked on top of each other. Okay. So cell culture, what is it? It's one cell is put into the tube of water with nutrients. Okay, the cell will divide and eventually develop roots. Okay, and leaves to grow into a new plant. So this is excuse me. So they do this with carrots a lot. You could really do it with any plant as long as you have the cell. So you take the root of the carrot plant. You have a cell cluster on the nutrients, and you'll put it in some water. It'll grow, and then your younger plants will transplant, and then they grow into a carrot. So tissue culture. A tissue sample from a plant is grown in an environment with special hormones and nutrients. So it is in a controlled environment. The tissue will eventually grow leaves and roots and grow into a plant. So this is how they get petunias, tobacco plants, and tomatoes most of the time. This is easier. It's an easier way. So what happens is you have a tissue sample straight from the parent plant. So you're taking the tissue, okay, so see where this is the knife, they're scraping that tissue. They're going to put it in a petri dish. The samples will grow into tiny plantlets, and then you'll transplant them. Okay, so after you watch the lecture, you'll have some questions. Make sure you post a picture of your notes onto the Canvas page, guys, okay? If you have any questions, send Ms. Wallace an email.